So, welcome everyone again to another episode of the ideaXme show that profiles the humans behind the really big ideas that are shaping our world, inspiring the future and future creators, and for all those that like great stories. I'm Ira Pastor, your a life sciences ambassador along for this journey. Today we are going to head back in the direction of Alzheimer's disease, this global scourge that's projected to affect over 100 million people globally by mid-century, and which is, as they say, truly a disease of substantial unmet medical need. We're honored today to be joined by a true rock star of this space, and a rock star on the keyboards in his own right. We'll get into that a little later. Dr. Rudolph Tanzi, who is the Joseph and Rose Kennedy Professor of Neurology at Harvard University, Vice Chair of Neurology, Director of the Genetics and Aging Research Unit, and co-director of the Henry and Allison McCance Center for Brain Health at Mass Gen, and who has been investigating the genetics of neurological diseases since the 1980s, when he participated in the first study that used genetic markers to find the disease gene for Huntington's disease. In 1990, Dr. Tanzi received his PhD in neurobiology at Harvard Medical School, where his doctoral thesis was on the discovery and the isolation of the first Alzheimer's disease gene, APP, amyloid precursor protein. Dr. Tanzi is involved in discovering all three familial early onset Alzheimer's disease genes and several other genes from neurological disorders, including those responsible for Wilson's disease. He currently serves as the chair of the Cure Alzheimer's Fund Leadership Group and the leader of the Cure Alzheimer's Fund Alzheimer's Genome Project, where he's carried out multiple genome-wide association studies for thousands of Alzheimer's families, leading to identification of a wide range of Alzheimer's candidate genes, including CD33 and ADAM10. He's also worked on the role of zinc and copper in Alzheimer's disease, on gamma secretase modulators for both preventing and treating Alzheimer's, and on human stem cells, ultimately to create sort of Alzheimer in a dish models and three-dimensional neurocultural systems. Dr. Tanzi has published over 500 scientific papers, including 3 of the most cited papers in the field of Alzheimer's disease. He has co-authored multiple books, including Decoding Darkness, The Search for the Genetic Causes of Alzheimer's Disease, the New York Times bestseller Super Brain, Unleashing the Explosive Power of Your Mind to Maximum Health, Happiness, and Spiritual Well-Being, Super Genes, Unlocking the Astonishing Power of Your DNA for Optimal Health and Well-Being, and The Healing Self, a revolutionary new plan to supercharge your immunity and stay well for life, co-authored with Deepak Chopra. Dr. Tanzi has made numerous television appearances on shows like CBS This Morning and Dr. Oz. He hosts the shows Super Brain with Dr. Rudy Tanzi and Super Genes with Dr. Rudy Tanzi, and the Brain Body Mind Connection with Dr. Rudy Tanzi and Deepak Chopra on PBS. And in addition to when he's not doing all of that and solving the dementia and Alzheimer's disease problems, he is an accomplished musician, playing keyboard professionally with Joe Perry, with Aerosmith. He's co-written songs, tribute to Alzheimer's disease patients called Remember Me. A lot to go into, a lot to discuss. Dr. Rudolph Tanzi, thanks for taking the time to come on the show.
Well, thank you for that very kind and generous introduction. Thank you for having me. It's a pleasure to be here.
Absolutely, absolutely. We typically start off the show by giving our guests the floor just for a little bit to talk about themselves. If you could take a few minutes, just for those sort of outside of the life science world, to introduce yourselves, or where you grew up, how you got interested in neurobiology, and ultimately how this domain of Alzheimer's and Alzheimer's dementia really became your area of focus for the last few decades.
Well, I've been at Harvard Medical School and Mass General Hospital my whole career, from student to professor. So it's been a long, long run. I grew up not far from Harvard. I grew up in Rhode Island, a tiny little state nearby. And I live just south of Boston in Milton. And, when I first got into science, I was working in the very first, literally the first ever project to find genetic markers. So today, people talk about SNPs and variants in the genome. Well, Jim Gasella, my mentor, who's a few years older than me at the time, in the early 80s, we found the first genetic markers ever, and then used those to try to find the gene for Huntington's, and then got extremely lucky in that, here's a genome 3 billion bases wide, right? So 3 billion rungs on a ladder.
Sure.
we picked up 5 random markers, right? So that's
Okay.
one base out of 3 billion that could be anywhere in the genome. And we found the gene. I mean, the odds against what we did were like 20,000 to 1, but we got lucky fast. And that kind of launched the whole need for the Human Genome Project. And it was the first time a disease gene had been found by genetic linkage. And, this was Jim Gasella's idea. I was the hands. I was at the time playing in a rock band 5 nights a week and, showing up to work, half asleep from playing too late the night before. But they were amazing days. And then I wanted to take that and I wanted to build the first map of a chromosome. Again, this is before Human Genome Project.
Sure.
So I wanted to build the first end-to-end map of chromosomes. So I picked the smallest one. As a student, you want to get done. So I picked chromosome 21, smallest chromosome. That led me to Down syndrome because they have an extra copy of chromosome 21.
Mm-hmm.
I learned that Down syndrome subjects get Alzheimer's pathology inevitably by middle age.
Mm-hmm.
So I just purely speculated there'd be an Alzheimer's gene. There was no Alzheimer's genes known yet. And I predicted it would be on chromosome 21, and that's why Down syndrome people get the pathology. And I further predicted it would make the plaques. We got the senile plaques.
Mm-hmm.
So it was tons of speculation upon speculation. And the higher ups at Harvard, where I was doing my degree, told me this was way too much speculation and risk for a grad student. But it was that kind of playing in a rock band mentality of, I don't care, I'm going to do what I want. I'm going to play the song the way I want. And it turned out, it's sure enough, the first Alzheimer's gene was on 21. It did make the plaques. All the predictions miraculously were true. So got kind of lucky there in the early days and been riding that now and just doing all kinds of different stuff. Aim is to cure Alzheimer's disease. That's the major aim.
Absolutely. An important name. Very important name. You obviously the area that you work in has been dominated for a while now with sort of the beta amyloid theme at its core in terms of the plaque formation, and then ultimately the neurofibrillary tangles, tau protein destroying synapses and so forth. But in your work, you've really, I use the word sort of the ecosystem of Alzheimer's disease. You've really approached this from a much greater perspective of looking at sort of everything that's happening here upstream and downstream. And I'd love to go into sort of each of these things one at a time. And I think one of the most fascinating, just because of the significant amount of activity going on in the area of the microbiome nowadays, is this principle of looking at beta amyloid as sort of this part of this more ancient innate immune system that we have, as a way that our brain tries to fight off forms of infection. You recently published a fascinating article entitled "Alzheimer's Disease Associated Beta Amyloid is Rapidly Seeded by Herpesvirus to Protect Against Bacterial Brain Infection" in the journal Neuron. I'd really like to learn more about, because we've talked a little bit on the show about the microbiome. To talk a little bit about how this area has sort of become more popular recently. I know that there's been some pushback from, I guess, other areas of the Alzheimer's space. Nonetheless, it is clearly an area that makes a lot of sense. A lot more groups beginning to look at it. And if you could also talk about sort of what some of the thinking is after sort of you validate this concept. Are we talking about antimicrobial agents? Are we talking about modifying the microbiome in some fashion? What type of potential interventions could we potentially see when we're dealing with this microbial response as part of the Alzheimer's pathology?
Well, I think we need to start with, if we go back to the 80s and 90s, the first 3 genes we found, and I was lucky enough to be involved with finding all 3 of those. The gene that makes the plaque itself, the amyloid plaque. So I named that amyloid precursor protein, APP. Right. And then we found what we call the presenilins. These are genes that make the enzyme that allows the amyloid to be made. So the enzyme cuts APP to make amyloid beta. Then after that, ApoE, the most common risk factor for Alzheimer's, the ApoE gene was found. And all 4 of those genes in the 80s and 90s said amyloid, 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 right? And it was George Glenner who discovered what amyloid's made of in 1984. We said maybe Alzheimer's is an amyloid disease of the brain.
Okay.
And then here we had the first 4 genes in the 80s and 90s, and the only thing they had in common was they lead to more amyloid in the brain. So it was perfectly reasonable for everybody to jump on the amyloid bandwagon and say, let's hit amyloid. Now we found the cause of the disease and we can cure it.
Mm.
So then you fast forward and there's all kinds of problems that come up, right?
Right.
You put amyloid genes in a mouse and they get amyloid, but they don't get the tangles that you mentioned. The tangles are what actually kill the nerve cell from inside. So why isn't amyloid causing tangles? And then there were all kinds of excuses made up. Like, oh, it takes a really long time to make a tangle and kill a cell, and the mice don't live long enough.
Mm.
So we lived with that fallacy for a while. Well, if you fast forward to the whole thing, it says, yes, if you get away from mice, because they're mice, they're not humans, and you put the amyloid genes in a mini human brain organoid in a dish, where you have human brain cells in a 3D matrix that, it's a facsimile of the brain the size of a paper punch, and it's 3D, then you can see for sure, yes, amyloid causes the tangles that kill the nerve cells. So that's great. But then why did all the amyloid trials fail? Every trial trying to stop amyloid did not make patients better.
Sure.
Well, then we learned the hard way, because that's what science is. You have to learn from your mistakes. As long as you're learning from your mistakes, you're doing good science. And then we learned that, hey, the amyloid, based on brain imaging, now that we can see plaques and tangles by imaging of the brain, the amyloid comes a decade or 2 before the symptoms. It's like cholesterol and heart disease.
Right.
If someone has congestive heart failure and just had a heart attack, the doctor wouldn't say, here, just take this Lipitor, just take statin to bring your cholesterol down. That's what causes heart disease. You had to do that a decade or 2 before. Same thing here. We learned the hard way, you have to stop amyloid early. And the amyloid causes the tangles, you have to stop the tangles early. Those are called the initiating pathologies.
Okay.
So there are 2 questions then. If amyloid and tangles come early and they initiate the disease, A, what's causing the amyloid and the tangles? What's causing the amyloid in the first place? And B, how do you get Alzheimer's disease if we stop amyloid in patients and they still stay demented? You don't help them cognitively. And that's where you're getting to with your question, which is, and so the first answer is, well, genetics and many, many different genes can allow some people to make more amyloid over time than others, just like cholesterol and heart disease. And on the other side of the coin, what actually causes dementia? That's easy. The plaques and the tangles cause just enough nerve cell death to trigger neuro inflammation. So just like with COVID-19, which we're going through right now, the COVID virus only kills X number of cells as a virus, but that triggers a immune response that involves inflammation. And then in the lung, you get what's called a cytokine storm. The cytokines are these chemicals that cause inflammation. Same thing in Alzheimer's. First, you get plaques and tangles, some neurons die, that triggers a cytokine storm in the brain of neuroinflammation. And that's when you get enough nerve cells dying to get dementia and disease. So that says 2 things. One is if you want to stop this disease, if you want to help patients, you have to stop neuroinflammation. Just like if you want to help a COVID patient, stop the virus early, but if they're already sick, you got to stop the inflammation in the lung. Second, if you want to nip the disease in the bud, well, for COVID, we're trying to get rid of the virus. And Alzheimer's, you have to stop the plaque from forming. And there, you're talking about drugs that slow down amyloid plaque. Or we could think about what triggers the plaque besides genetics. And that's where my colleague, Rob Moyer, who tragically passed away in December last year with a glioblastoma after only a 3-month battle with the disease, 58 years old. Terrible thing. Rob was literally my best friend, and I lost my best friend. But Rob suggested, hey, maybe microbes like viruses and bacteria are triggering plaques to form, because he noticed that the amyloid beta of the plaque is antimicrobial. So at the origin, you have maybe genetics and infection driving plaque formation. And then that causes tangles to form. Now we know that. The mice misguided us. And that kills X number of nerve cells. Then that triggers inflammation. And then you get the widespread neuronal cell death that causes dementia. At that point, if you want to help a patient, you got to stop the neuroinflammation.
Mm-hmm.
There's a forest fire going on. It's too late to blow up the match, which is why all these trials have failed against amyloid. So that's right there about 35 years of history of Alzheimer's in a nutshell.
And when it comes to the neuroinflammation, is this, and once again, I think about inflammation, we hear this term inflammaging nowadays, how inflammation is so important in all diseases, whether it's CNS, cardiovascular, and so forth. Is this something that you see, I mean, obviously comes to mind in anti-inflammatory substances, but then on the other hand, we do need some level of inflammation. Is there, what is being thought of in terms of some of the different, once again, going beyond sort of these amyloid antibodies, what's being thought of in terms of neuroinflammation? Is it unique anti-inflammatories that get in the central nervous system? Are they maybe immunomodulating agents? Like, what types of things do you envision as sort of the interventions for this type of very unique inflammation?
Great question. Yeah. So first, we have to distinguish inflammation in the body, in the lung, in my knee that I played way too much basketball on. That is different inflammation.
Okay.
Right. What they have in common is inflammation is meant to protect you, right? Inflammation occurs whenever there's tissue or damage to a cell or tissue. Our immune system, our innate immune system, so there's the innate immune system, which protects you right away with inflammation. And there's the adaptive immune system, which takes its time to make antibodies and mount, B cell, T cell interactions, antibodies. Adaptive immunity is more for the long-term protection, memory B cells. But when you first get an infection, it's your innate immune system that keeps you alive. There are antimicrobial peptides.
Okay.
That's what amyloid beta protein is. That's what Rob discovered in my lab. There are macrophages that go and eat and attack. Well, you got to remember that, it wasn't so long ago, 10, 20,000 years ago, that humans were running around the jungle. Our lifespan was 25 years old. And we were living in complete squalor, right? We were filthy. And we were getting infected by things all the time. We lived like one big infection. So our innate immune system that causes inflammation evolved to say, these are filthy animals who need to constantly be taken care of. They're getting cut all the time and bacteria are getting in. And so inflammation was made to protect us against our lifestyle. But now, fast forward to where we are now, we live hygienically. There was a scientific revolution. There was an industrial revolution. We covered the sewers. We have antibiotics. Well, guess what? The innate immune system never got that memo. They have no idea. The macrophages, the microglia in the brain, they're still like, cells are dying, guys, wipe it out. There's a cut over here and it's bleeding, wipe it out. So 20,000 years ago, somebody's running around the jungle and nerve cells start dying in the brain. That wasn't because of Alzheimer's. Lifespan was 25 years old. He got like some super mosquito bite. He has like some Zika or West Nile or whatever else is going around, causing meningitis or encephalitis. So the inflammation system in the brain evolved to say, neurons are dying, wipe it out. Now, fast forward, you're 70, 80 years old, plaques and tangles are accumulating for whatever reason. And I want to argue that they evolved to accumulate to protect us, because the plaques can protect us, the tangles can protect us. We'll get into that. But now the plaques and tangles are accumulating and certain numbers of nerve cells are dying. And now the glial cells in the brain, what are called microglia, astrocytes, they see the neurons dying and they say, whoa, wait a minute. They're on the same exact alarm clock as they were 20,000 years ago. Neurons are dying, wipe the area out, wipe it clean. It's compromised. They don't know that it's due to plaques and tangles. They don't know it's not due to a Zika infection. So neuroinflammation, in essence, is our evolutionary baggage in our brain. And now it doesn't allow us to live long enough such that our brain span, our brain health span keeps up with our lifespan. So you have to understand how neuroinflammation happens in the brain before you can treat it. And I'm going to tell you how we're going to treat it.
Okay.
But we had no idea until 2008. So in 2008, we discovered the first Alzheimer's gene in our lab that causes neuroinflammation. It's called CD33.
Okay.
And CD33 is simple. On these little microglial cells that are usually like little cleaning cells during deep sleep, they eat amyloid debris, they clean up, they nurture, but they always are looking around for nerve cells dying. And when they see nerve cells dying, they switch from being housekeepers to terrorist killers. And they're like, whoa, too many neurons died. That wasn't amyloid I just ate. That was a membrane of a neuron I just ate. We're in trouble here. Wipe the area out. That's neuroinflammation. CD33 is the on switch for that to happen. And the next gene found was TREM2, TREM2. That was the off switch. So between 2008 and 2012, we discovered, and TREM2 was discovered elsewhere, we discovered CD33, but the on and off switch for whether microglial cells are benign housekeepers or nasty killers were discovered. And then we said, now we know the target. Now we understand what's turning on inflammation in the brain after plaques and tangles form and a certain amount of neurons die. Now we can learn, we can think about how to screen for drugs to control it. And, you know, I'm happy to share. That's the next chapter I could share with you if you don't mind.
No, definitely. I mean, if you can, I'd love to hear a little more about that. That's such an important part of the puzzle.
Yeah. So what we had to do was we had to modify the first. We made this Alzheimer's in a dish model, you know, where you're growing a mini human brain-like organoid in a dish.
Okay.
So now we had to add. We originally modeled it to make plaques and tangles. This was Duyan Kim and my group and others. So now we had to add neuroinflammation to the dish. Add these little glial cells, microglia, astrocytes. We did that, and because of that, we could now, you know, each one of these mini brains is the size of a paper punch. They fit in a well, in a plate this big in a 96-well dish. So now what you can do is rapidly screen every approved drug, every natural product you can find, to say what stops the plaques, what stops the tangles, but most importantly for helping patients right now, what stops the neuroinflammation. So you can use these genes like CD33 to turn on the neuroinflammation and then say what helps. And to get to things that stop neuroinflammation, there are two ways you can do it. So if you have somebody shooting people up in a crowd, right?
Right.
There's two things you can do. You can give bulletproof vests to the people being shot and protect them, or you could take out the killers. And that's exactly the type of drugs we got, right? So some of the drugs we found were neuroprotective. While neuroinflammation is going on, and they kill by causing what's called oxidative stress, they deplete the neurons of energy. Then you can try to protect them against that. The other thing you can do is take these microglial cells that are killing and find drugs or products that convert them back to being housekeepers that are benign. So now to explain where we went with this, I have to describe 3 companies in which I have equity. I'm a shareholder, and for conflict of interest and transparency, I have to say that before I get into what they do.
Absolutely.
One of the natural products that came up was a woody vine from Peru called cat's claw.
Okay, sure.
And in Peru, going back thousands of years, they would grind up this powder from cat's claw, and they would take it to help the immune system and help against infection. And so we published a paper with Alan Snow, a colleague of mine with whom I started a company called Cognitive Clarity, and showed that it hits plaques, tangles, and neuroinflammation, albeit no clinical trials were done. So we cannot make any claims about helping Alzheimer's, because until you do a clinical trial, it's just not responsible to say that.
Sure.
But in a mouse model, at least, it was effective against Alzheimer's type pathology. And that, Alan has in the company it's sold as a supplement called Percepta, which is cat's claw plus an antioxidant from tea. So that's for neuroprotection. On the drug side, I worked with a couple of guys who, when they came to see me, they were undergraduates at Brown University. And they were in a fraternity called Sigma Chi. And this was 2013. And when I was in college, I was in Sigma Chi. It's a national fraternity. And that year, I had been put into the equivalent of the Sigma Chi Hall of Fame. And so they read my name and they saw I was doing Alzheimer's, and they were doing an undergraduate project on Alzheimer's, and they had an idea for how to protect neurons against neuroinflammation. And we worked out that idea together, and it looked like it had legs. Next thing you know, we started a company. The company is called Amylyx, A-M-Y-L-Y-X. And Amylyx just announced in December that their trial to stop neuroinflammation in ALS, Lou Gehrig's disease, worked. So this is a neuroprotective drug combination that was able to slow down ALS. And now that paper is so the details haven't come out yet, just the fact that the trial work came out in the press. But the paper describing those results is under review at New England Journal of Medicine, hopefully be out soon, and will be formally announced. The results will all be announced. And the company's discussing approval with the FDA. But it just shows you how fast things can happen once you hit the right target.
Absolutely.
So in other words, if you were trying to hit these pathologies that initiate the disease, like plaques or tangles and Alzheimer's or Lewy bodies and Parkinson's, or the inclusions that form inside of neurons and ALS, you have to get there really early. Because the symptoms are happening when there's already neuroinflammation triggered, and you're killing 10 to 100 times more neurons than what the initial pathology you're killing to trigger neuroinflammation in the first place, right? So that's basically the common theme. And anybody who finds a drug that works against neuroinflammation in one disease, like Alzheimer's, is probably going to work in Parkinson's, Huntington's, ALS, frontal lobe dementia, because they all have the same profile, that the initiating pathology in certain area of the central nervous system determines what's going to go wrong, memory or movement. But it's not until you trigger neuroinflammation that you actually get enough cell death and synapse loss to get to the symptoms. And I like to say all the time, the elephant in the room in our healthcare system is that we don't diagnose neurodegenerative disease until the brain's degenerated to the point that it can't function correctly. And imagine if we did that in diabetes.
Sure.
Imagine if we said, no, we're not going to treat you until you've lost enough islet cells from your pancreas. And when your pancreas is properly degenerated, then we'll try to treat you. Or heart disease. No, we're going to wait till you have congestive heart failure. Then we're going to try to treat you. And we're going to give you a drug that stops cholesterol. Oh, wait a minute. You needed that 10, 20 years ago. So, brain disease is just catching up. If you want to hit plaques and tangles, you have to do that with early detection, early intervention, decades before symptoms. If you want to help patients right now, you have to stop what's killing the neurons. That's neuroinflammation. And that's what, I just gave you two examples of how we do that, more toward neuroprotection. The third company I became involved with is called AZ Therapies. And again, for transparency, I have equity in these companies, so there is conflict of interest as I discussed them. But AZ Therapies, we found when we screened, we found that this drug called cromolyn, it's an old asthma drug.
Okay, sure.
That hits mast cells and stops mast cells from being activated.
Yeah.
Well, it turns out the same drug can chill out these microglial cells that have been activated. So here's these microglial cells in the brain that go from housekeeper to killer. And in the screen, we found that cromolyn calms them down, so they stop killing and they go back to being housekeeping. They're clearing amyloid again. So what we found is that if you add cromolyn to microglial cells in the dish, the Alzheimer's in a dish, that it takes these killer microglial cells that are eating neurons and axons and synapses, and then turns them back into housekeepers that are now cleaning up the amyloid around the neurons. So that drug is in a phase 3 trial for Alzheimer's disease, and will read out later this year. And I want to also warn that you can't take the cromolyn used for asthma and use Alzheimer's because it won't get into the brain, okay? So the regular cromolyn asthma thing just goes into the upper respiratory. The company had to make a new formulation of cromolyn with a new delivery system that gets it into the deep lung, into the blood, and into the brain. So I don't want people running out using all cromolyn inhalers for Alzheimer's. You have to have the right formulation and delivery. But that will read out. So AZ Therapies Alzheimer's trial will read out later this year. They're just starting in ALS. Amylyx did an ALS trial first. Now they're doing an Alzheimer's trial. Again, if you're hitting neuroinflammation, either from protection against it or stop it, you can really try it in any neurodegenerative disease that threatens healthy aging.
It's a fascinating set of approaches, and I mean, two of the interesting themes. I mean, it's all very interesting, but the concept of sort of this repurposing. Cromolyn is, as you said, it's been around for maybe a century or so. I forget how
Yeah.
long.
Oh,
Here's a very elegant example of the things that we have already. Maybe we don't need to synthesize anything new, but we have a lot of these very important pharmacologic moieties that can do other things. And then the cat's claw thing is really neat because, this, as we talk more about sort of the combinatorial components of this disease, these plant extracts and so forth, they're mixtures of bioactive substances that can do more than one thing. So it's very nice to see that some of this sort of more ancient knowledge can be, once again, repurposed for something like this. I want to sort of transition a bit into aging, which is we discussed the theme of sort of healthy longevity, health span, and so forth on this show. And obviously beyond your Alzheimer's work, you've done a lot of work with some of the movers and shakers, Deepak Chopra, Dr. Oz, and so forth, in sort of the aging field. You have the book Super Brain, where you go into these concepts of what I'll refer to as sort of psychobiological. How about everything that goes on up here is not just a couple pounds of stuff inside the skull, but can impact our thoughts, our feelings, can impact everything via the endocrine system, lymphatic, affecting everything, our organs, our tissues, our cells. Talk for a little bit about, I know you focus on spiritual stuff as well, but talk to a little bit about where we are, in your opinion, with regard to sort of this psychobiologic nature of aging and disease today.
I think the first thing to remember is when we say psychobiological, is that psycho is biological.
Okay.
Right? So there's the spiritual side, which we can put aside, and there's the mental side, and there's the physical side. But what people don't often think about is that a mental disorder is a physical disorder. Okay. And growing up, I used to hear, someone who had a mental disorder had a neurochemical imbalance. Like everything's fine in the brain. Everything's fine, except, those neurochemicals just aren't balanced right. That is downstream. That's a symptom of what's going on. The fact is, in depression, in schizophrenia, there's inflammation as well, right? There's physical stuff happening in the brain that's killing neurons or compromising synapses in neurons. And then, depending on which neurons are being attacked, you'll get various symptoms because those neurotransmitters, those neurochemicals aren't going to be made correctly. So the neurochemical imbalance that causes the so-called mental disorder starts with a physical defect, a physical alteration that was caused by genetics, or we argue microbes, right? We have found that plaques can be triggered overnight by bacteria and viruses, overnight. That tangles inside the nerve cells can be triggered overnight. The plaques try to trap the microbes. Once a herpes virus gets into a neuron, how does it travel? It travels down the axon across the synapse. Well, guess what tangles do? They block it. It's a roadblock. So is it any surprise that when the plaques try to trap the virus and some virus gets away, next thing you know is the plaque plus the virus triggers a tangle. So now it can't get down the axon. And then on top of that, the amyloid decorates the nerve cell and says to the microglial cell, eat this one, get rid of it. It's infected. So that's why we say that plaques, tangles, and inflammation are part of a orchestrated innate immune response to protect the brain. And this mattered 10,000, 20,000, 30, 40,000 years ago. But over time, genetics arose. So every big bout and epidemic of brain encephalitis, meningitis, the people who survived had gene mutations where they made plaques more readily. They made tangles more readily. They were able to trigger neuroinflammation faster. They survived the big Zika and the big meningitis and encephalitis outbreaks for the last 50,000 years. Then we carry those genetics today, and what happens, you try to live till 80 years old without Alzheimer's, and the people who have these genes that were conserved trigger the disease. So we're not saying that microbes trigger the disease today. Maybe in some cases they do, but it's a combination of the genetics we carry with us because this pathology evolved to protect the brain. That's this innate immune protection hypothesis. So now, let's bring that now to brain health.
Okay.
We need to figure out a way to keep our brains healthy our entire lives and not wait for disease.
Mm-hmm.
Well, think about when you go to a doctor every year. What do you go for when you go every year to the doctor? What do you call it? You go for your what?
My annual checkup.
Or your annual
Physical. Sorry.
You go for a physical, right? From the neck up, what do? They look in the holes in your head. Nose, mouth, eyes, ears. Oh, there's something in there? Yeah, we don't know how to look at that. We just kind of skip over that. This is a physical, not a mental. And the reason is, yeah, you could do a neuropsych exam, but what good is that going to do for a 30-year-old? The reason is we don't know how to quantify brain health. We learned how to quantify heart health. We have stethoscopes, we have blood pressure, we look at cholesterol, we can look at insulin and glucose for diabetes. For neck down, we figured out how to look for signs of disease decades before it comes and causes symptoms. But from the neck up, there's no checkup. As Patrick Kennedy likes to say, there's no checkup from the neck up. So we started a center at Mass General called the Hemi and Allison McCance Center for Brain Health. And our goal is not to treat patients, but to treat people who are well or at high risk for disease and be able to learn how to quantify brain health. So when you do trials to keep the brain healthy, you know what to look for. So we're looking for biomarkers in the blood. We're looking for ways to image the brain. We're looking ways to combine that with EEG. I started a company, again, equity. I started a company called React Neuro, where we make a VR device that is assessing eye tracking, voice analysis, and neuropsych testing all in one, and an algorithm that combines that to say, can we get you a brain health index? You know how like when you get and this came up because the other thing I do on the side is I work with, because I do watch football, NFL football. I feel guilty sometimes because, these guys are, for our entertainment, are risking their future lives by getting so many bangs to the head. So when I had a chance to give back and was asked by Bill Belichick and the New England Patriots to revise on brain health, I took the chance. And one of the things that Coach Belichick asked, he said, I don't want to send a guy back onto the field if it's not safe and they had a concussion. How do we know when it's really safe? Is there a way to objectively quantify that? And I said, no. Best thing you got is follow my finger with your eyes, and let's see if your eyes track smoothly. So what we developed a device to do that originally, and then we figured out that, hey, maybe with age, over time, over decades, as it turns out the same pathology that accumulates in the brain when you get banged in the head that can cause what's called chronic traumatic encephalopathy, or CTE. It's tangles, just like Alzheimer's. It's inflammation. But in this case, you don't need the amyloid to cause the tangles. It's the head bang that causes the tangles. So Alzheimer's disease is an amyloid-induced tangle disease that causes inflammation and symptoms. CTE is a head bang-induced tangle disease. So think about it. These head bangs are coming in your teens and 20s, causing pockets of tangles, cell death, inflammation. It takes decades of that pathology to spread before you get symptoms. Same thing in Alzheimer's. So what we figured out is that we can track this age-related pathology of plaques and tangles and inflammation over time by looking for subtle changes in the eye movement. So if you're a neurologist and I'm saying follow my finger, I need to be, I need to see problems with your eye tracking that are in, seconds or maybe microseconds. But if you have eye tracking issues that come with pathology slowly over age, it may be nanoseconds. So that's why we had to develop a technology that can track changes in eye movement at a nanosecond rate, so that over time of aging, could it be that this same pathology that so acutely affects you during concussion and your eyes can't track it all, is it happening more gradually and insidiously with age, and we need a device that's more sensitive. So this is another way to track this. This is another way to get to the point of a stethoscope or a blood pressure cuff for the brain. Blood biomarkers, imaging, EEG, digital devices combined with neuropsych testing, voice analysis, eye movement. This is all the type of things we need to do to assess brain health. Now, how does that get us to spiritual side, the psychobiological? Well, my last book I wrote with Deepak Chopra, The Healing Self, was to talk about the incredible influence of your mindset, your perspective, your thinking on your health, because there's an incredible link between the brain and the immune system.
Yep.
And you can talk yourself sick, you can think yourself sick, you can think and talk yourself well. And that's called Psychoneuroimmunology.
Okay.
And it's the most mysterious field there is. But there's a there, okay? You know, all woo-woo science is woo-woo until you learn how to measure it and predict it, then it becomes real science. But when people criticize me and say, why are you looking into this woo-woo stuff? I say, because that's where science begins. You know, the metaphysical becomes physical once you can measure it and predict it. That's what do we do as scientists? Shut up and measure. Learn how to predict. You can't measure until you know what to measure, and you can't measure it until you know how to measure it. But you have to think about how to do that. So right now we're spending a lot of time saying, how do we measure the effect of the brain and thinking and mentation on the immune system?
Mm-hmm.
Once you figure out how to measure it and predict it, it becomes a science. Now nobody makes fun of you anymore. But it has to start somewhere. So that's why I'm so willing to engage in writing books like Super Brain and Super Genes and Healing Self, to say, where might we go? What do we know now? I bring the science, Deepak brings the spiritual side, and say, what might happen in the future when science and spirituality meet? Because that's where all great things come from.
Absolutely. Absolutely. While we're still on that subject, you just reminded me of something. I might as well just throw it in now, because a couple of weeks ago, I did a show with Sharon Inouye up there at Harvard on sort of the topic of delirium, and one of the sort of the side themes that popped up was this sort of this bizarre occurrence in much later stage Alzheimer's patients known as terminal lucidity, where memories somehow reappear in some way very late into the disease. I'm just, when you're talking about some of this stuff and sort of the unknowns of what's inside there, have you ever done any study on some of that type of stuff, and any clues on, there's also the issue of how Alzheimer's patients can still know the music, but they'll forget someone's name. Any of these sort of unknown things that are interesting to you that you spend a little time on as well when it comes to dementia?
Yeah, I mean, terminal lucidity is just the biggest mystery. Even delirium's a mystery. I mean, why do people, when they have a urinary tract infection, get delirious? Is it the bacteria from the urinary tract that's getting into the brain? Is it something the bacteria make? Is it an immune system in the body, immune inflammation system in the body that affects the brain? We don't even know that. We know so little about delirium. And in Alzheimer's with dementia, I mean, yes, I mean, how people suddenly can snap into having memory. I think you have to think about the mechanics of memory. It involves synapses, right? So if every day of your life while you have Alzheimer's, your immune system in your brain, your very primitive immune system in your brain. I like this, Rob Moyer, my late colleague, used to say, the brain's immune system is at the same level as a horseshoe crab or an oyster. Okay. And so the immune system's constantly attacking neurons and synapses and eating them and attacking them. It's really hard to think while that's happening. But like we wrote in Super Brain, neurons are always trying to compensate. If two neurons are next to each other, we know that if this neuron dies, the next door neighbor neuron will immediately somehow know how to regain the synapses that were lost from the next door neighbor. Compensatory regeneration. How the heck does this neuron next door to this guy who dies say, oh, I got you covered, and finds the same connections again? It's crazy, but it happens. That's why we can recover from strokes. We can recover from TBI, brain injury, compensatory regeneration, plasticity. That's why we wrote Super Brain. But the question is, we know, well, the fact is we know so little about how this works. We know so little about how many synapses do you need to be able to recall a memory. And when you can't recall a memory, like a name or have lucidity, is it really because you don't have the neurons to do it? Or is it the same as the mystery of delirium, where somehow the brain is clouded and then it clears? With delirium, you can have a peripheral, like I say, urinary tract infection, and that clouds the brain so you can't think straight and you're delirious, and all of a sudden the bacterial infection clears and whatever got into the brain clears. It's like, oh yeah, I'm all right now. But what really happened was inflammation stopped. So I think what happens with these patients is toward the end, there is such a huge intentional, mental boost to say, whoa, I'm leaving now. I need to know what's going on, that it's maybe some type of psychoneuroimmunological event where you overcome that neuroinflammation. And suddenly with the synapses you still have, the clouds clear and you can think. Scientifically, what I'm saying is complete BS. It's woo-woo. I don't know the mechanism. I'm just saying what might happen. I have no idea how. In science, if you can't explain how, then you're not doing science. Like, we don't explain why. Why is for philosophers. We do what, where, when, and how. We stop there. But, but I think if we had to really study it, I think somehow in those last days, the neuroinflammation somehow clears through intention. I don't understand how, but it's a great topic for investigation.
I hope you do a book on that.
Well, the thing is that as a scientist, we only have one job as a scientist. We measure things, we predict things, and based on that, we go to try to disprove things. That's what we do. Nope. When I have students who are trying to prove a hypothesis, it's like, not in my lab. Your job as a scientist is to come up with the best idea you possibly can this morning in the shower, and then go into the lab like it's the idea of your worst enemy and trash it and kill it and see if it can stand up. That's how you do good science. And as long as you're going to think in that mindset of disprove your hypothesis, trash it, kill it, that gives you the license to think freely about all the possibilities, unlimited, about what might be happening at the boundaries of spiritual, mental, and physical existence. You're allowed to wildly extrapolate to absurdity as much as you want, as long as you know that when you bring it into the lab, you have to know how to measure it or figure out how to measure it, predict it, and then when you test your hypothesis, aim at killing it, crushing it, and seeing if it holds up. Then you find that balance between being a visionary and actually getting to the truth. There's people who don't do that, it causes some problems, right? The people who want to just talk about fantasy and say it's already science, like pseudoscience, or people who just want to have a really good idea and want to just go to prove it, and they convince themselves something's happening, but their data doesn't hold up for 5 or 10 years. That's the thing, is you want to make discoveries that are going to hold up for 50 to 100 years. That's the goal, at least 50 to 100.
Excellent. I have to ask you, you wrote an article a couple of weeks ago with Dr. Chopra. It was entitled, "What is the best self-care during the COVID-19 crisis?" Obviously, you mentioned cytokine storm is a major part of this. You can cytokine storm in the brain with Alzheimer's, and then obviously there's been a lot in the press with regard to this disease. It kills the lungs. It does other things in our bodies now, vascular system, in our CNS. That being said, any advice, nutrition, meditation, or otherwise, in terms of how the average person should be taking care of themselves?
So what prompted that was in the last book Deepak and I wrote, The Healing Self. We talk about chronic systemic inflammation. That as we get older, we all have some baseline level of inflammation that's chronic and systemic throughout the body and the brain. And a good part of the book talks about the science behind that. And then the second half of the book is, what can you do about it? What can you do in your life to try to reduce? And all the advice we give, again, until you do clinical trials, you can't say it's going to work. You can only say, here's the empirical evidence that says it's worth a try, right? So you have to, again, just like with science, you have to be careful when you talk about clinical treatment. If you haven't done a trial, don't make any promises. So I'm not making any promises here.
I understand.
Because for most of the stuff, trials have not been done because people just don't do these trials. There's no money in it, right? There's no money in telling people to get more sleep. Unless you have a product that helps them get to sleep, I guess. But anyway, at some point, this is a crazy story. So the book came out, January 2018. And that same month, a new album I had done with Joe Perry from Aerosmith,
Yep.
Johnny Depp, and some others. Ringo Starr's son was on drums. Zach Starkey plays for the Who. Great. It's an album called Switzerland Manifesto, because Johnny Depp lives on Switzer Street, and the studio is called Switzerland.
Yeah.
And so this album was coming out the same week as the new book, and I had to do both. Had to be involved with both, and so for the book, I said, man, I need a quick way to like just summarize this, what we call the 7-day action plan for bringing inflammation down.
Mm-hmm.
And I was in the shower, and I keep a pad in the shower called Aqua Notes.
Okay.
And the pencil says, don't let those good ideas go down the drain. Because, you know, like when you're in the shower, you get the hot water on your head, and you kind of go into a meditative, relaxed state. Mm-hmm. And, you know, like in the shower, you get all kinds of amazing ideas, and you kind of like, you know, in and out of, you know, you're kind of going from alpha beta to a theta-like state. Mm-hmm. And then you get out of the shower and you towel off, and you're like, what the heck? That was so good. What was I thinking about? What was I talking about? You know, it's all gone, right? Because that theta state's gone. Now your mind's racing back up at alpha beta. So the pad allows you to quickly capture it. It's like these rubberized notes with a pencil that writes on the note. So I had this idea of SHIELD, how to keep your inflammation in your body down with SHIELD, or shield your brain. And S is for sleep. So you need to get enough sleep to clean your brain. It's during deep sleep after REM, your brain cleans all that crap out. Those little microglial cells that can cause inflammation are normally housekeeping, and it's just after REM during deep sleep that they actually clean out the amyloid and debris. So it's how you clean your brain. I jokingly call it mental floss. Okay? And then H was handle stress, meditations. Deepak and I actually published a paper showing that a meditation practice actually reduces inflammation at the genetic level. This is a paper that had, you know, Elizabeth Blackburn, Nobel Prize winner, who discovered telomerase. She showed that telomerase levels were reduced. Telomerase levels were increased, which is good. When we have a meditation practice, we saw a 40% increase in telomerase. Alyssa Appel was on this paper. Eric Schott, who's a very famous geneticist, did the genomics with us. So we had an all-star team, and we found clear evidence that meditation at the genetic level and the level of telomerase, and, you know, which builds the chromosome tips, were positively influenced by meditation. So handling stress, meditation, and there is genetic evidence for that now. I mean, there is biological evidence for that now. Interaction, I is interaction, social interaction. I remind people that during COVID right now, it's physical distancing that we're experiencing, not social distancing. We're on Zoom with our friends, and, you know, we're probably interacting more than ever on Zoom and on, you know, all kinds of social media. Even though we're physically distanced, social interaction is still possible. So interaction, loneliness is a risk factor for Alzheimer's. Okay. Loneliness increases stress. Stress induces inflammation. So handling stress is the H, meditation, I, interaction, social interaction. E is exercise. We published a big paper a year or so ago showing that exercise induces the birth of new neurons in the brain, neurogenesis. It also helps clear inflammation, so physical exercise. L, learn new things. So if you're learning something new, you're making new synapses, you're building your synaptic reserve. At the end of the day, Alzheimer's is loss of synapses. I mean, as you know, when everything goes wrong, inflammation, et cetera, it's how many synapses you lose that causes dementia. And so the more synapses you make and in reserve, the more you can lose before you lose it, right? So you want to learn new things all the time, not brain games, learn new things. Like right now, people watching your show are learning new things. Sure. Unless I'm putting them to sleep and I'm still helping them because
learning new things.
Right. And then D is diet. Diet meaning Mediterranean diet, #1 for the brain. You know, that means no meat, more fish. I'm vegetarian, so I don't eat fish either. But, you know, feeding your microbiome, probiotics, prebiotics, lots of fiber, nuts, seeds, grains, vegetables, and probiotics. So your microbiome, your gut microbiome directly affects the health of your brain. There's the gut-brain axis. We actually published several papers showing that we could make Alzheimer's mice better by actually manipulating their gut microbiome. So that's it. So that's SHIELD: sleep, handle stress, interaction with others, exercise, learn new things, diet. And you have to shield with extra intensity right now, so that so you can attempt to keep your baseline inflammation down. So if COVID hits, you already have your base level down. No guarantees, no clinical trials done, right? But why not?
Yeah. Sounds sensible, and hey, it's taking advantage of things people have been doing for a long time. So,
yeah.
Final question. I just have to ask you, any, obviously, you don't have any gigs coming up, but anything planned in terms of post-COVID, new albums, concerts, any big events you're playing at?
I text with Joe. Joe Perry is become a very close friend. Joe Perry, of course, being co-founder of Aerosmith, the guitarist, one of the best guitarists alive today. And so when Joe needs keyboards, he often uses me for studio keyboards. I don't have time to gig or tour. They have Aerosmith has a great touring keyboard player who can sing, unlike me, Buck Johnson, who's great. Sure. So Joe and I talk all the time about, wanting to get back into the studio and play again and at least jam. And we're waiting for COVID to pass to do that. Another good friend is Richie Sambora, who you may know. Richie Sambora is the founding lead guitarist from Bon Jovi. And actually, I have to show you. What we're trying, the neurology residents have all been on the front lines of COVID lately. So they may even have to defer the next stage of their career because it's all hands on deck, and they're not going to be able to have a graduation. So what we did was we asked various people if they can make videos for the virtual graduation, and Richie Sambora made a song for them. I'll show it to you.
Oh, that's
He just sent this to me last night.
That's awesome.
With my words, just don't seem enough. Just want to thank you for saving us.
So that was pretty cool. So we're gonna be showing that at graduation. Joe did one, Maria Shriver, even Bill Belichick did one, and putting together these videos to thank them for their service in the front lines against COVID, and to congratulate them. So that's the music I'm doing these days. and I am playing a lot of piano, mostly taking requests from friends like jazz standards and recording them and sending them out by text. I have my own music website. I don't want to seem self-promotional, but it's numberonemusic.com/Rudy Tanzi. I have about 36 original songs there that are either solo piano or trio stuff, and I just do all my music there, and it's all free live streaming. So I keep it up.
Very cool. Yeah. Very cool. Well, Dr. Tanzi, it's been a real honor having you and sharing your endless knowledge with us, and really, you had an amazing career, but really wishing you the best in taking a lot of these next step forward in this very, as this scourge that everyone's saying is going to be the major issue that we're going to be dealing with this century in terms of chronic disease. So really thanking you for that, what you've done and what you're going to continue to do. For everybody that's going to be watching on the YouTube channel or listening on the podcast networks, you've been listening to the amazing Dr. Rudolph Tanzi, Joseph Rose Kennedy Professor of Neurology at Harvard University, Vice Chair in Neurology, Director of Genetics and Aging Research Unit, and Co-Director at the Center for Brain Health at Mass Gen Hospital. Check out his numerous books on Amazon, his music website. Try to follow the SHIELD protocol to get us through this COVID-19 period. Really amazing stuff. Thank you, Dr. Tanzi, for coming on the show. Thank you for doing everything you do. And as we say in the show, thank you for moving the human story forward with your work. It's really amazing stuff.
And I would thank you for having me, and thank you for moving the human story forward, for all the hard work you do to bring these stories and bring so many people's stories to everyone in the world. And so thank you for all you do.
Thank you for that.